Hi, everybody. I say everybody so casually, and then I have to stop for a minute and really think about what that means in today's virtual world reality. Now, everybody refers to people who literally are all over the world. And just so that you know, we have people signed up from all over the world: Lithuania, South Africa, Australia, Canada, and over 40 different US cities. How amazing is that? That's huge. And actually, it's as huge as the content of this evening's unique event. I'm Pamela Nathan. I'm the president of SAJAC, the South African Jewish American Community, and we're so proud to be presenting our very first virtual symposium, and hopefully there'll be more. Initially, SAJAC was started in Los Angeles by the late Dennis Abramson. Our chapter, the only surviving chapter, was started by Leah Levine in 1987. And big kudos goes to Leah. She surrounded herself with an amazing group of Sapphirs, as they refer to us in South Africa, who've continued to live up to her vision of having a meaningful social organization where people of similar backgrounds could identify with one another while establishing themselves in a new environment, and at the same time preserving our rich Eastern European heritage that preceded our immigration. And this is still relevant today, 33 years later. Let me tell you that our annual Reporter magazines, dating back to 1987, have recently been archived by the San Diego Jewish Historical Society. And currently, Dr. David Klein, and not my nephew, David Klein, is doing an oral research project on our South African Jewish Ashkenazi community in Southern California. This David, spelled C-L-I-N-E, is a professor of history at San Diego State University. So if you want more information about this or anything else about SAJAC, what we do, how we operate, we'll be in the 5th breakout room in the 2nd hour of our symposium. So please do drop by and say hello. Happy to answer any questions you may have. Membership is available nationwide now. You know, SAJAC is renowned for organizing unforgettable, fabulous social events. Big dance parties, braais, exhibitions, award galas, and of course, they're usually in person. But this year, due to the impact of COVID, there was nowhere else to go except virtual. And so it was. And we decided on this extremely important topic, and we thought that it would be very well received. And I'm happy to see that it really is. And so a big thanks goes to all of you for showing up today. And now it's time to introduce you to Dr. Franklin Galis, who we invited to help to coordinate our symposium. Franklin got his medical degree at Wits, graduating with an MBBCH cum laude. Then he emigrated and completed his general surgical and urology training in the US, as well as 2 years of basic science research. Today, he serves as Chief Scientific Officer at Genesis Healthcare Partners here in San Diego, and he's also a voluntary professor of urology at the University of California in San Diego. Franklin, thanks so much for all that you have done to bring this amazing group of experts together. And now it's over to you.
Thanks so much, Pam. Good evening and welcome, everyone. It is a great honor and pleasure to act as the moderator of what we know will be a most informative symposium and provide you with useful information. Many thanks to Pam Nathan and the SAJAC board for supporting this event, as well as our sponsors, Myriad Genetics, Invitae, Sharsheret, and JScreen. We're joined by several amazing and talented people who are generously sharing their time and personal stories to help educate us understand this complex and important topic. How might our Ashkenazi inherited genes potentially impact our health? I personally find this to be a fascinating topic which interconnects our Jewish history, science, technology, and medical advances, all of which may have bearing on one's health. On that note, Judaism teaches us about the sanctity of life and a most sacred mitzvah, pikuach nefesh, the obligation to save a life. If just one person's life is improved or saved as a consequence of this symposium, this would be most rewarding. So let's get started. This symposium would not have occurred without the tremendous help of multiple people, and I'd like to acknowledge them. Firstly, Pam Nathan, SAJAC president. Pam has been the president of SAJAC for many years, probably decades. She's the glue who's kept this organization alive, the last SAJAC standing in the United States. So thank you, Pam. Kudos to you. Tonight, we are privileged to have Dr. Karim Kader, a dear friend and colleague, co-founder of Stratified Genomics, professor of urology at the University of California, San Diego, a nationally renowned urological surgeon scientist who has a deep interest and passion for genetics and has developed tests for detecting prostate cancer risk. We're joined and we are very fortunate to have two wonderful people who will generously share their personal stories. Sarah Abelson, whose late mom Sandy, of blessed memory, who fought breast cancer courageously. Sarah will share her personal perspective as a BRCA carrier. And I just want to personally thank Sarah, who is 8 and a bit months pregnant, for sticking with us and being here today. Thank you, Sarah. Clive Vesico, a dear cousin of mine, a great friend who will share his personal perspective on prostate cancer and genetic testing. Our sponsors, Myriad and Invitae, who are leaders in the field of genetic testing, Sharsheret, a national organization supporting Jewish women and families of all backgrounds facing breast and ovarian cancer, those who are diagnosed and those who are at risk. And then JScreen, a national nonprofit public health initiative dedicated to preventing Jewish genetic diseases. JScreen is headquartered in Atlanta at Emory University School of Medicine. They offer carrier screening for over 200 genetic diseases that could impact a couple's future children, as well as follow-up genetic counseling services. Special thanks to Charlene and Sean, our IT support folks, who are making this symposium feasible, and the SAJAC board members who are going to be hosting the breakout sessions. The breakout sessions are available and we encourage you to participate. The first breakout session is the breast session. So please, during the course of the symposium, feel free to sign up for whatever is of interest to you. You can move from one breakout session to another. The way to get to a breakout session is to go to the chat room, click on everyone and go to support, and they will put you into the appropriate breakout session. The first breakout session, the breast session, will be hosted by Dr. Linda Smith from Albuquerque, a renowned breast surgeon and expert on genetics, together with Sarah Abelson. The second breakout session will be the prostate session, hosted by Dr. Karim Kader and Dr. Stacy Loeb, a colleague who's a world-renowned prostate cancer researcher from New York. And Dr. Loeb will be joining myself with Clive Essico. The genetic counseling third session will be hosted by Peggy Cottrell from Sharsheret, and the carrier screening session by Emily Goldberg from JScreen. And finally, as Pam mentioned, the SAJAC Informational Center. So please feel free to join the breakout sessions. I'd like to just disclose that Dr. Kader and myself have a relationship with Stratified Genomics. And please note that the materials and opinions presented this evening are reflective of the individual presenter's opinions and not necessarily of SAJAC. The presentations are intended for general education and not meant to serve as medical advice or any specific medical recommendations. So who are we, Ashkenazi Jews? The Ashkenazi Jewish community is thought to have arisen in Europe, Germany, and France between the 6th and the 9th century CE. Ashkenaz in Hebrew refers to Germany, and recent genetic analysis suggests that we are actually a mixture of Central European and Middle Eastern ancestry, and we're actually a distinct single genetic community. Today's 10 million Ashkenazi Jews descend from a population of only about 350 individuals who lived about 600 to 800 years ago. And about 80% of modern Jewry worldwide have Ashkenazi ancestry. I think it's important to get a historical perspective to explain how our genetic makeup landed up the way it is today. And it starts around 1095 with the first Crusades, of which there were 5. Persecution in Western Europe led to migration east. And in this map, taken from the Atlas of Jewish History by Sir Martin Gilbert, and I'd encourage anyone who wants to learn more about our history and migration, it's a fabulous book. But what you see here is a map indicating Germany, France, where the Crusades start. Initially, there was violence and expulsion of Jews from all the communities highlighted in black background. And over 200 years, several crusades, the Jews, our families migrated east towards Poland and Greater Lithuania. And what you see here is the map of Eastern Europe. And the reason we landed up here, the Grand Duke Vitov and Casimir the Great welcomed Jews and actually developed protective decrees that Jews would not be harmed. And they were welcomed because of their education, their mathematical skills, their business skills. And ultimately, the Pale of Settlement was established by Catherine the Great in 1791, where Jews, and I highlight, were permitted to reside. We eventually were not welcome in the major cities, and so we lived in little towns scattered throughout the Pale, which includes much of present-day Lithuania, Belarus, Poland, Latvia, Ukraine, and parts of Western Russia. And Jews were heavily congregated in these little towns, which we know as shtetls. My wife Gina and I visited family shtetls back almost 20 years ago in Latvia, and we noted that the Ludza shtetl we come from was 40% Jewish. So in summary, the makeup of the Jewish population, 80% Ashkenazi Jews originate from the Russian area, Eastern Europe, and Western Europe. In contrast, Mizrahi Jews are from Egypt, Saudi Arabia, Iran, Iraq, and Sephardic Jews originate from Spain, Libya, Turkey, and Syria. And this all has impact on our genetic profile, which is explained by what is called the founder effect. And this refers to a high frequency of a specific gene mutation in a population founded by a small ancestral group. So if we look at the left-hand box, the original population, you see there's one individual depicted in orange who has a genetic mutation, and all the folks depicted in blue have normal genetic makeup. And then we move towards the right, and you see a significant attrition of the population, a decrease in the number, and the concentration of the one person with a genetic abnormality is much greater now. So, as I've explained below, the marked population decrease together with a bottleneck where the Jewish people were restricted, and with other factors like genetic isolation. We are typically a somewhat closed community in that we historically have married other Jews, resulting in a closed gene pool. And another factor, consanguine marriage, which is marriage between individuals who are closely related, which unfortunately happened in past generations, all contributed to our decreased genetic diversity. And then what happens later, you land up generations later with a much more concentrated number of people. You can see the orange dots reflecting a much increased number of individuals who carry these genetic defects. And this is where we are today, unfortunately, where 1 in 40 Ashkenazi Jews carries the BRCA1/2 mutation defect, compared to the general population, 1 in 400, and that's a 10-fold increase. If we look at carriers of autosomal recessive disease, such as Tay-Sachs, cystic fibrosis, Gaucher disease, spinal muscular atrophy, and many more, we Ashkenazi Jews, 1 in 4, are carriers of an autosomal recessive gene mutation. What's interesting is to see that we are collectively very distinct from a genetic signature standpoint. And here is a genome-wide genetic signature of Jewish ancestry, which perfectly separates individuals with and without full Jewish ancestry in a large random sample of European Americans. So I bring your attention to this plot here, and you see on the right-hand side a concentration of Ashkenazi Jews, and if you look at the key, all 4 grandparents are Jewish. Now, when you move to the left, you have Ashkenazi Jews where 3 grandparents are Jewish, in yellow 2 grandparents Jewish, in green 1, and then the general population who are not Jewish. So you can see Ashkenazi Jews are very distinct according to their genetic signature. And if we look at ourselves compared to other Jews, looking at Iranian Jews, Italian Jews, Syrian Jews, Greek Sephardic Jews, Ashkenazi Jews are very distinct again. So our genetic signature is quite different from many others. I'm going to move into some brief introductions on basic cell biology. Dr. Cater is going to teach us a lot more in a short while. This slide just serves to explain what's going on in the genetic code. Here is a cell. Each one of us is made up of about 50 trillion, not billion, trillion cells. Our bodies are made up of trillions of cells. And within each cell is a structure called a nucleus, where the genetic code is housed in the form of chromosomes. We each have 46 chromosomes, and within each chromosome is this long-stranded, what we call DNA, and please note the double helix, which becomes relevant in the next slide I will share with you. So I thought I'd share today a genetic timeline to give you a perspective of where we are today with the incredible knowledge that we can test for BRCA1 and 2 genetic mutations. We've now got drugs to treat patients who suffer from malignancies related to BRCA1 and 2 mutations, and how far we've come to get this knowledge and develop these therapeutics over just over 150 years. And we'll start off with Gregor Mendel, who was a friar and considered the founder of the modern science of genetics. And he discovered the fundamental laws of inheritance. And for those, and I know there are a few youngsters in the virtual audience, he didn't have sophisticated models and technology. He used peas, and he looked at the different colors of peas and figured out what we understand in modern day genetics in 1865. In 1903, Baveri determined that there was a relationship between chromosomal abnormalities in cancer. And then if we fast forward to about 1952, the famous Watson and Crick pair who won the Nobel Prize for establishing the double-stranded DNA helix, which was fundamental to our understanding of DNA and genetics as we know it is today. And then I'm just going to fast forward to the 1980s. What's relevant to us as a community, and many of us, I'm sure, remember the days we were tested for Tay-Sachs. When my wife Jean and I got married in the 80s, we were tested for one gene mutation, Tay-Sachs disease, which was discovered in 1980. Currently, today, young people getting married are tested for 225 Ashkenazi genetic-related diseases. I'll just bring you back to the late 1950s. Trisomy, which causes Down syndrome, was identified, and which almost all pregnancies are tested for today. And then finally, let me just say a word or two about the Human Genome Project, which was completed just 17 years ago, which was the complete deciphering of the human genetic blueprint, sequencing DNA, the entire genome, which has just created an opportunity to learn so much more of our genetic structure. As one can see in this publication from the New England Journal of Medicine in 1997, the risk of cancer was associated with specific mutations of BRCA1 and BRCA2 in Ashkenazi Jews. And I emphasize the point mutations and the association with breast, ovarian, and prostate cancer, which we've more recently appreciated. But it's important that we should all remember that we all carry BRCA1 and 2 genes, and in their normal state, they help actually regulate cell growth. They repair DNA, which is happening all the damage, which is happening all the time, and suppress development of tumors. And mutations to either gene can result in uncontrolled cell growth and can lead to the development of malignancy. That's why we now test certain populations for BRCA to intervene before this happens. And let me bring us back to where we are today, COVID-19, unfortunately, the pandemic, which has led to this virtual symposium. And borrowed from Scientific American, genetic engineering could make a COVID vaccine within months rather than years. To quickly create potential vaccine against COVID-19, researchers are using the tools of genetic engineering, and this contrasts to the traditional methods. And I just highlighted the polio vaccine, which we're familiar with, was first attempted in 1935, subsequently and successfully utilized by Dr. Jonas Salk, who had the Salk Institute, and there's a Salk Institute in San Diego, who developed the first successful inactivated vaccine. So I greatly appreciate your attendance and your patience in listening to this presentation. And now it's my distinct pleasure to turn over to Dr. Karim Kader for his talk. Thank you.
Can everybody see my screen?
Great, yes.
Wonderful. Well, Franklin, thank you so much for your kind invitation for me to give this talk today, and thank you, Pam. And thank you to all the volunteers, the patients, the control individuals who've helped further our genetic studies over the years. I've got an ambitious set of objectives for this evening. The first is really to gain an appreciation of how amazing our life is, understand the role of DNA, genes, and chromosomes in natural biology, get a better understanding of how cancer is truly a genetic disease, understand how genetic predisposition can lead to prostate cancer, and understand what role genetic markers can have in future prostate cancer care, not just screening, but care. Now, it's difficult for sometimes to remind ourselves, especially during these divisive times, but life is truly a miracle. By just being here, we've all won the biggest lottery in the world. When you imagine that one sperm fertilized your mom's egg, it could have been one of the 150 million other siblings, but that's the one that got in, and you're here today. Not only that, we rely on trillions of cellular divisions to go from the single cell stage, that fertilized egg, to the fetus that was born. These are trillions of flawless divisions, and the immensity of this will become clear as we understand that DNA is really the secret code of life. Now, DNA is a sugar that has 4 different base pairs: guanine, cytosine, adenine, and thymine. These are just 4 different codes that human biology uses to make protein. So DNA codes for protein. And here you can see an example of the entire genome, starting with the base pairs to the double helix that Franklin eloquently discussed earlier, all the way into the chromosome. There are 3 billion base pairs that need to get replicated with every cellular division. 3 billion that need to get transcribed completely over each cellular division. Now, the working unit of the human genome is the gene, and it's the gene that encodes for the protein that is ultimately delivered. But there's only 20,000 genes in the entire genome. So 20,000 genes, 3 billion base pairs, it shows you that the majority of DNA is what we call junk DNA or non-coding DNA. We don't really understand what it means, so we call it junk. So Franklin mentioned inherited traits. We get 23 chromosomes from our mom, 23 chromosomes from our dad, and ultimately we end up with a fertilized egg that subsequently develops into an embryo. Now, Gregor Mendel was the grandfather of genetics, and he first described this concept of inherited traits, of dominant and recessive traits, and how they kind of contribute to the next generation of phenotype or trait. So genetic mutations, by definition, are very rare events. They occur in about 5% of the population. They typically involve genetic change that codes for protein. And they're strongly associated with disease risk. That means that mutation results in a change in the trait. And that's something called penetrance. So a high penetrant mutation leads to a change in phenotype or trait. For example, BRCA1 and 2 are highly penetrant mutations. Now, just by way of example here, let's look at an autosomal dominant pattern of Mendelian inheritance. A lot of big words to say, this is how a mutation gets passed on to the next generation. On the right-hand side here, we see an unaffected mother with 2 normal copies of the BRCA2 gene, which exists on chromosome 13. Here we have an affected father who has 1 copy of a mutated BRCA2 gene. And you can see as a result, by only having 1 affected chromosome, he has a 50% chance of passing that on to the next generation. So 50%, those individuals in white here have inherited that mutation, 50% did not. Now, single nucleotide polymorphisms are another form of genetic change that can predispose individuals to malignancy. These are the single base pair differences between me and you. And that Human Genome Project that Dr. Galis had mentioned earlier was the first glimpse we had into this. Now, there are 300 of what these called SNPs in the human genome. And some of these are associated with disease risk and some are associated with cancer risk. They exist in everyone. And they're essentially what makes me, me, and you. Otherwise, 99.9% of our genome are identical. Each individual SNP is not highly associated with disease, so these markers are called low penetrant markers. But when we put a bunch of these SNPs together, they turn into excellent markers for disease risk. Interestingly enough, and as you'll see later on in the talk, they're not associated with family history. Unlike BRCA2, for example, which is an autosomal dominant type of pattern. Now, the oncogene was first appreciated with the Philadelphia chromosome in 1960. This was the first example of a genetic mutation that was associated with disease risk, and this was chronic myeloid leukemia. Since then, there's been dozens of mutations associated with cancer in general, including the BRCA1 and 2 genes, as Franklin alluded to before. These are the genes responsible for repair of DNA that has been damaged thanks to environmental exposures. Now, it's important to note that cancer is a genetic disease, and it's an accumulation of genetic change, which ultimately leads to the dysregulated growth that we see with cancer. So you can imagine how, if we're not repairing these genetic damage that occurs with time, well, this DNA damage can just propagate, and a vicious cycle ensue that ultimately leads to cancer. Well, let's dive in and talk a little bit about prostate cancer. Now, this is a very personal disease. We all know people who've been affected by prostate cancer, and unfortunately, we all know many men who've been, unfortunately, succumbed to the disease. It's the most common solid organ malignancy affecting men, and it's either the second or third most common cause of cancer-related death. We know we can cure it when we catch it early. However, there's a conundrum. Sometimes we don't even want to diagnose men with cancer, with prostate cancer, because it's typically a slow-growing disease, and for many, the cure is worse than the disease itself. Now, why do we screen for prostate cancer? Well, symptoms are extremely rare until it's incurable. It involves regular digital rectal examinations, which neither the doctor nor the patient enjoy, but are important. In addition, it includes a regular prostate-specific antigen or PSA blood test. So, PSA. PSA screening has worked. We've seen a tremendous decline in the incidence of metastatic disease at presentation, from 70 per 100,000 down to about 25. The same cannot be said for screening mammography. So clearly PSA has had an important role in screening. But unfortunately, PSA has not been the huge hit that we were hoping for. So screening everyone for prostate cancer is extremely controversial right now. So many have turned to this concept of targeted screening, risk-adapted screening. So focusing our screening efforts on those individuals who are at highest risk. And we know that the risk of prostate cancer really focuses on family history, race, and age, with family history being a very important one. So when we look at prostate cancer inherited risk, there can be a family history-related risk, or there can be genetic differences, such as mutations in BRCA2, as well as single nucleotide polymorphisms. Now, focusing only on individuals with a family history is good, but it's not enough. If we were only to focus on individuals with a family history, we would miss out on 80% of cancers. People without a family history are still at a substantial risk, or could still be at a substantial risk of disease. And family history is dependent really on the number and age of family members, as well as the prevalence of screening in the society. So back in 2005, the Genome-Wide Association study was initiated, and that coincided with my graduation from fellowship. And together with a gentleman by the name of Jianfeng Xu, we worked to develop a single nucleotide polymorphism test, which is a stable objective measure of a man's lifetime risk of prostate cancer. Since then, over the last 15 years, we validated the test in over 100,000 men, and really it's indicated for all men of Caucasian race, and now we can say Hispanic, and we have an African American test that's about to come out, and we do have a Japanese test as well. That is simple to interpret and acts very much like a family history. Now, what about the BRCA2 gene and the risk of subsequent disease? Well, we know that in men, there is a tremendous increase in breast cancer risk. Most people don't know, but men can have breast cancer. So it's important for men to understand that risk. Prostate cancer risk. It is up, but it's not crazy high. 1.5, I would argue it's probably closer to 2 times the lifetime risk of the average American man, which is about 11%. Pancreatic cancers are also at higher risk. But the implications for women are probably more important, with breast cancer at 11 times, ovarian and uterine at substantially higher risk. So let's focus on 2 brothers, one of which we're going to meet tonight. These are Caucasian immigrants from South Africa, both of whom are healthy. They are urged by their cousin, who is a very prominent, very handsome, very charming man, to undergo PSA-based prostate cancer screening starting at age 50. Now, their father died at age 50 from heart disease, so they don't have that same opportunity to understand their family history. Their cousin was diagnosed with prostate cancer in his 60s, and their younger brother began testing after learning of his brother's diagnosis. Okay, so here's a chart of their PSA exams or studies. So brother A, okay, who we will meet later on this evening, had a steady rise in his PSA from 2005 and 2010, and he was urged to get a prostate biopsy earlier in time. He eventually got one in 2010, which demonstrated some low-grade prostate cancer. He underwent a prostatectomy and is doing very well today. Now let's compare this to his brother. His brother had fluctuating PSAs, and this can be a real problem in men with PSA bouncing up and down. But you can see that he had 1 negative biopsy, a second negative biopsy. And this obviously is an uncomfortable thing. And this is something we'd like to avoid if possible. Now, I alluded to this SNP-based test that Franklin and I are working on. It's called PROMPT, or the Prompt Prostate Genetic Score. And you can see that brother A had an extremely high PROMPT test correlating with his prostate cancer diagnosis in 2010, whereas brother B had an extremely low result, suggesting that these PSA fluctuation may be due to benign causes, such as a benign prostatic hyperplasia. Okay, so the actionable information that could have come from this type of testing would be that for the older brother, you could have considered an earlier biopsy and an earlier diagnosis of cancer, whereas the younger brother could have at least avoided the second biopsy and maybe even the first biopsy, given his low score. So it's clear that genetic testing is going to play an important role for prostate cancer care in the future. But it is important not to overextend what these results mean. So when getting any form of testing, it's important for us to consider what we would do with the results to make sure that it's actionable information and to understand the limitations and implications of testing. So, for example, for BRCA2 with men, the risk of prostate cancer is high, but it's not to the point where you should be losing sleep. Or there are some ways that we can talk about screening more aggressively and potentially even decreasing your risk of disease. So the bottom line is we're entering into a very exciting era where this type of testing can help guide not just our diagnosis, but our management of disease. Franklin, again, thank you. I did run over a little bit. I apologize. But I'm open to any kind of questioning that you may have.
Karim, thank you so much. That was magnificent, a tremendous overview. Just a pleasure to have you part of the panel. It's given me and everybody else tremendous insight into genetics and the role it plays and what can be done. And now it's my great pleasure to introduce a dear cousin, Clive Essico, who will speak on his diagnosis of prostate cancer from his perspective. Thank you, Clive.
Thanks, Franklin, and thanks to SAJAC for giving me the opportunity to share my story with all the participants who are present at the symposium today. I was diagnosed with prostate cancer approximately 11 years ago at the age of 54. I'd like to give a little of the background leading up to this diagnosis. Just prior to turning 50, my cousin and good friend Franklin, who spoke a short while ago, urged me to pay his office a visit so that he could give me a routine urological checkup. A close relative of mine had been diagnosed with prostate cancer, and it was clearly prudent for me to commence having regular checkups. The examination revealed no issues, and I had a PSA test which came back with a reading of 1.1, certainly nothing to worry about. I scheduled annual visits with Franklin, and each time my PSA results came back higher. The consistent upward trend became cause for concern, notwithstanding the fact that my PSA was still very low. My PSA at the age of 53 was 3.12. However, when tested the following year, the reading again was higher, this time 3.3. While a reading of 3.3 was nothing to be worried about, Franklin was concerned about the continued consistent increase over time. A digital examination yielded an area of what I recall was being explained as uneven texture of the prostate, and I agreed that Franklin perform a biopsy. 12 cores were extracted, and to my shock, after all, I was only 54 years old at the time, 5 of these were positive. I had prostate cancer. The positive samples were located in 3 of the 4 quadrants of the biopsy map. And so if you look at just a sort of a oval type picture, it was spread all about the prostate. At the time in 2010, prostatectomy was by way of robotic surgery was in its infancy. And I accordingly elected to have a radical prostatectomy by way of a long incision made through the abdominal wall, starting at the belly button, going all the way down. Due to our close relationship, I didn't want to place pressure on Franklin to do the surgery. And so the surgery was carried out by an extremely experienced surgeon at USC in Los Angeles. The most traumatic part of this journey, apart from the initial shock, was waiting to hear the pathology results, which would indicate whether the prostate capsule had been penetrated, meaning that the cancer had spread outside of the prostate walls. The surgeon advised that he personally would let me know the results of the pathology, and he would either pop into the ward or call me telephonically as soon as he received these. I started playing mind games, and my prayer was that I would hear the phone ring rather than seeing him appear in my ward, as the latter would indicate a more severe diagnosis. Fortunately, the phone rang the following day, and he gave me the wonderful news that the margins were clear. About 10 years later, I learned of the discovery of a test alluded to by Dr. Cater, the prompt PGS test, which could determine a man's genetic predisposition to prostate cancer. By way of a simple mouth swab, a genetic test could provide a score indicating one's lifetime risk to prostate cancer. I was fascinated by this, even though it was after the fact, and so carried out this non-invasive test myself. As expected, my results reflected that I had an above average risk of prostate cancer. I asked myself the question, had I had the benefit of the results of this test in or about 2010, Would I have done anything different? Firstly, knowing my results, I wouldn't receive the shock of the diagnosis. I would have expected that at some point in time, realistically, I may have prostate cancer. Further, while I'm extremely fortunate that I had a vigilant and thorough urologist, perhaps I would have had a biopsy earlier, which would have mitigated against the risk and reduced the agony of waiting for the results of the pathology by having diminished mental anguish, even to a slighter degree. I would have lessened that burden. The other benefit of the prompt PGS test was that my brother Steve, who had a far higher PSA reading than I did, as explained by Dr. Cater, and who had already been biopsied twice, and anybody who's had that biopsy with a needle going somewhere you don't want it to go, twice, due to his very high reading, could have been given an indication as to his risk and perhaps avoiding those biopsies. Particularly now, knowing that his brother had prostate cancer, he was extremely concerned. Was he genetically predisposed to the same disease? My brother took the test and fortunately was found to be of low risk. And so while he continues to have regular checkups, this has been a great relief to him. And so the bottom line is, with the incredible advances in medical science and technology, I urge all men to have regular prostate checkups, consult with their doctor, and take that prompt PGS test, and in this manner, be an active participant in their future health and welfare. Thank you again for allowing me this time, and I'm more than happy to make myself available for questions during the breakout session.
Clive, thank you so much. Greatly appreciate the insights and sharing your personal experience. Wonderful to have you on the panel, and so generous and kind of you to share your personal experience. It's now my great pleasure to bring back Sarah Abelson to share her experience. So let's have Sarah up, please.
Good evening, everyone. Thank you for joining us. I'm here to share my personal experience with the BRCA2 genetic mutation. When I was born at about 2 years of age, when I was only 2 years old, my mother, Sandy Lewis, was diagnosed for the very first time with breast cancer. At that time, we didn't know how severe and how long this disease would endure and how long she would battle it. And she ended up battling the disease for over 18 years until I was 20 years old. She unfortunately lost her life to breast cancer. So throughout my entire life, breast cancer and BRCA have been terms that I'm overwhelmingly familiar with, I'm knowledgeable about, and I've been very educated on the matters. I always knew what the risks were, and throughout my mom's battle, I got to watch her fight fiercely and really endure every 2 years when she would have a reoccurrence, and just see firsthand how this disease can really impact somebody's life. As mentioned, every 2 years she would have a reoccurrence, and eventually, about 8 years prior to her passing, we found out that she was indeed BRCA2 positive. At the time, it was still very new what BRCA meant. People were just really coming to terms with the implications and what this knowledge really serves to others, and all the different risks involved. Immediately, my mom only had one sister who was also tested. She also was BRCA2 positive and took matters into her own hands and did the prophylactic mastectomy. Throughout my entire life until the age of 20, I always had in the back of my head that at some point I would be tested and I would have to deal with whatever those results might be. When my mother passed away a few months after, my dad and I spoke, and I said, I think it's time for us to know what my future is. To me, knowledge is power. I'm empowered by knowing what my options are, whether it was young or not. I think when you know the results of what lies ahead, you're more eager to really dive into it and know what you need to do. So at age 20, I tested positive for BRCA2, and I said right then and there to my dad, at some point I'm going to do the preventative surgery. I think because I had such a close interaction with breast cancer all of my life, for me, the preventative mastectomy was a no-brainer. It was just a matter of when and where. I always thought it would be something that I would do after having kids, after settling down, and a fairy godmother of mine said to me one day, you know, you might want to think about doing it a little bit earlier. Luckily for me, I was blessed with meeting my husband at a very early age. I was blessed getting married and having the privilege of being able to do the surgery quite young. I did the surgery when I was 25 years old in New York City. After one weird biopsy, I just turned to my husband and said, I never want to go through that again. It's time we do the surgery and let's have it be behind us. It was 6 months after we got married, and I remember going into the date of my surgery with my husband and my dad by my side and just looking at them and saying, this is for all of our future. If we have the choice to leave BRCA and to lower my risk, this is where like our new chapter begins. Little did I find out that 4 months after my recovery and undergoing the preventative mastectomy, which I'm available afterwards for any questions. I'm happy to thoroughly go into more details about what that entails, because that can also come with an overwhelming amount of questions and knowledge, and it's a whole task in itself, and I'm happy to be there for anyone who's going through that. When I went for my follow-up checkup 4 months later, the doctor who did my preventative mastectomy turns to me and goes, okay, now you have this behind you. Have you ever heard of PGD? And I was like, oh God, what is that? He goes, so very recently, if you're interested in really eradicating BRCA from your life and starting completely with a new chapter and doing something really strong and wonderful for your family, you should look into how you can prevent BRCA with IVF. And as soon as those words slipped from his mouth, I was already Googling what that meant that evening. I made the decision 3 months later to start the process of IVF to do PGD, which means pre-implantation genetic testing. Through PGD, and like Franklin has mentioned, when you start to have a family, Jewish couples especially are at higher risks for over 200 and so genetic diseases. For me, I wanted to eradicate BRCA from my offspring completely. So when I started the process of IVF, before you can even start everything, your embryos are tested for your specific mutation, which in my case was BRCA2. And it turned out that out of the 15 embryos that we fortunately, my husband and I created, over 60% of them had BRCA, leaving us with only 5 BRCA-free embryos. When I heard those results, I was in a state of shock, because you don't even, it's one thing to do something for yourself, and for me to make the decision to do the preventative mastectomy, it's another thing to really put yourself in a position to do something for your offspring. And I also felt that I was doing something on a larger scale for everyone. If you have the power to eradicate BRCA, even in this small sense, it just was such a humbling moment that really made me feel like, wow, this is something, I'm so glad that we did this. So we had 5 BRCA-free embryos, and I'm very proud to speak here this evening to say that that I am about 3 weeks away from giving birth to a BRCA-free baby girl, which really, I think, brings everything we've spoken about full circle. Everything we talk about in genetics and how it connects to your offspring is really important because we're impacting future generations. And we have not only the power to educate and be knowledgeable about what our decisions can do, but how they impact those around us. And I would just like to say thank you for taking the time to listen to my story. I hope that it empowers others to get tested and to not be afraid to ask those really hard questions, because it's a very overwhelming topic that comes with a lot of dimensions and it never seems to end. But I'm here to tell you that if you want to change your future, if you want to know how to at least change the course of your future, you can. So thank you for listening, and thank you to Franklin and SAJAC for putting on this amazing event.
Thank you so much, Sarah. I mean, what more can I say? I'm looking at the chats. All I'm hearing is amazing, amazing, amazing. What a way to close off a wonderful symposium. I just want to thank everybody again for participating. For all of you viewers out there, I wish we could see you, but we know you're out there. It's been an absolute pleasure. I just want to see if we have time. I don't believe we've had some phenomenal pieces of information shared with us, both didactic and personal. And I think what we should do is move to the chat rooms where I ask you please to keep your questions. I'll be in chat room 2 with the prostate session, Sarah and Dr. Smith in chat room 1 for breast, and in genetic counseling, we've got terrific experts, genetic counseling, Sharsheret, Peggy Cottrell, and in chat room 3 and chat room 4, Jay Screen, Emily Goldberg, who will talk about carrier screening for those of you who've got questions about family planning, what tests to have done. So again, thank you all out there virtually for participating. We have had this symposium recorded. So if you feel that anybody might benefit from listening to these amazing people who shared their stories, Pam will send out information how to reach the link for the recording. So I'm going to ask Charlene to open up the chat rooms, please. And thank you all again.